Okay, so I'm going to post a video today kind of just explaining the Hershey and Chase experiment. So I asked you to go over 3 experiments. Griffith's experiment, excuse me, the transformation principle. Okay, he really didn't prove or even provide evidence that it was DNA and not protein transferring genetic information. But what he proved was that you can transfer information from one organism to another through some kind of molecule. Okay, we don't know whether it's protein or DNA yet. So Griffiths kind of, although it didn't definitively say it's DNA, he kind of provided an experiment that other people could use, and that's what Oswald Avery did. So Avery went through and he provided pretty strong evidence that it is DNA and not protein. And then Hershey and Chase basically completely solved it. They said, okay, it's definitively, without a doubt, it's DNA and it's not protein. Okay? So I'm going to kind of go through just a little bit of an explanation, but that's what we need to know. We need to know how they proved that it was DNA, not protein, and you need to know those 3 experiments. Okay? So let me go through Hershey and Chase's experiment a little bit more in depth. So here's Hershey and Chase. Okay, they were a husband and wife team. Now, during that time period, it should be noted, okay, and especially you'll see this during the Rosalind Franklin or the Photo 51 video, women weren't thought of as equals. Okay, it was a horrible time because they didn't think of women as equals, and it really held back our science. We could have gone a lot farther in science if we would have realized that men and women are equal, and that we can both do tremendous things in science. All right, so there's Hershey and Chase. So again, their big problem was they had to figure out, is it DNA or is it protein? So they went about this a little bit a different way. They were more of had a chemistry background than less necessarily biology. And they said, okay, well, we know there's 4 macromolecules, right? Protein, DNA, lipids, and carbs. We've already eliminated carbs and lipids. So we know it's either protein or DNA. Well, we know that protein and DNA are both made out of these molecules or these elements. Everything has to be made out of chenops. So they looked and said, well, is there an element that's in an amino acid or a protein? So this is the structure of one amino acid, okay? And then proteins are just polymers of amino acids. So here's an amino acid, and here's a piece or a nucleotide. So they said, well, is there any elements that's in protein that's not in DNA? And they said, oh, sulfur. Sulfur's in protein, but it's not in DNA. And then likewise, they said, well, is there any element that's in DNA that's not in protein? And they said, well, phosphorus is. So that's how we can differentiate between the 2 molecules. We can say if there's sulfur present, we know that it's protein, and if there's phosphorus present, we know that it's DNA. So they used this idea to basically formulate their experiment. So along with this understanding, they also used a very other handy thing called the bacteriophage. Now, you'll learn about these. If you go into biology, you're going to learn about these a lot more. But it's simply a very simple virus. Okay, now, not all viruses look like this, but this is one type of virus. We call it a bacteriophage. We use them all the time as basically kind of little syringes to put DNA in. So here is the virus, and it's simply 2 things. A protein outer coat. So the outside protein coat will have sulfur in it because it's made out of protein. And then the inside is where the DNA is, and that will have phosphorus in it. Okay? Now, the other thing that should be mentioned is that you can remove the DNA from this virus and you can put in other DNA. We'll talk about that when we develop GMOs and transgenic organisms. Okay? The key thing is the outer coat is protein, and then the inside is DNA. So what they're trying to figure out is if we're going to pass this genetic information, you're going to have to insert something into the cell. So here's the bacteria cell here. So what's going inside the cell? Is it the DNA that's going to go in, or is it the coat that's going to go in? Now, it seems pretty obvious to us looking at this that it's obviously going to be the DNA. But at the time, they didn't know if the protein coat was being absorbed by the cell and the DNA was staying out of the cell, or whether it was the DNA going inside and the protein staying on the outside. But they know whatever goes inside the cell must be the hereditary molecule, whether it's DNA that goes inside or protein that goes inside. So they did 2 little experiments. Okay. The first thing they're going to do is if we radio label sulfur. So what I mean by radio labeling, we don't need to get too in depth about this, is basically creating an isotope of sulfur. And kind of remember, isotopes have varying numbers of neutrons. And this isotope of sulfur is going to emit radiation. So basically, wherever there's protein, there's going to be an emission of radiation. So you can measure that radiation and find out, well, where exactly is the protein? Because wherever it's emitting radiation is where the protein is going to be. So they basically radio-labeled sulfur, and then they're going to So once we've radiolabeled, we can follow protein and see where exactly does it go. Does it enter the bacteria or does it stay on the outside? If it enters the bacteria, that would indicate protein is the genetic molecule. But if it stays on the outside of the bacteria, never actually enters it, that would indicate that protein isn't it. All right, so that's experiment 1, and we'll see the results in a second. And then we go to experiment 2. And they said, okay, well, let's run a completely different experiment in which we radiolabel phosphorus. So now we're going to radiolabel the DNA that's inside. We're not going to label the outside coat, but just inside we're going to radiolabel the DNA. And the same kind of thing, it's going to emit radiation. So we can follow DNA as to wherever it goes. Does it go inside the cell or does it stay on the outside? Right. So knowing that, now let's make sense of this mess. Okay? So we're going to start up here with our first experiment. And in this experiment, they radiolabeled protein. Okay? And for convenience sake, they said it's yellow. So right now we have protein labeled, DNA is not labeled. Okay? So we're going to then we're going to mix the bacteria. So here's a bacteria cell, and then here's the viruses. We're going to mix these together. Okay? And then we're going to blend it. Now the whole purpose of blending it is just to kick off anything that's attached to the outside of these shells. They didn't know what was attached on the outside, whether it was DNA or protein. But they knew they had to blend it to kick these things off so that nothing's going to be attached to the cells. Okay? Then we run a centrifuge. And a centrifuge is just used to basically kind of separate different molecules or different things that would be inside this blender. So at the very bottom of the centrifuge is where all the bacteria cells are going to go. They're the heaviest. And when you centrifuge something, the heaviest things go to the bottom. So at the very bottom, which we call the supernatant, you don't need to know that, but that's what it would be called, right, is a whole bunch of bacteria cells. So they're all sitting at the very bottom of it. Now, if protein, which we radiolabeled, actually went inside the bacteria cell, then at the very bottom where all the cells are, we would see radiation shooting out this bottom right here. But we don't. We see radiation shooting out at the top where the cells aren't. So that indicates that the protein never actually entered the cell. Because if it did enter the cell, it would be emitting radiation out of the cells at the bottom, but it's not. It's emitting radiation over the top. Okay? So that indicates that protein never actually entered the cell, which indicates protein's not the genetic molecule. Okay? So they said, okay, that's pretty convincing, but we need to, be sure. So let's run another experiment. So then they ran a separate experiment. And they said, okay, let's radiolabel DNA. And they go through the same thing. We radiolabel DNA. We're not going to radiolabel the protein. We go through this. We got to centrifuge it to kick off whatever is on the outside. And again, they don't know whether it's protein or DNA on the outside. And then we centrifuge it, and then all the bacteria cells are going to go to the bottom. Now, when they looked for what was emitting radiation, they noticed that it was at the bottom where the radiation was coming out. So that means the DNA that was radiolabeled somehow got inside the cell. It was inside the cell, and we could prove that because inside the cell was where the radiation was being emitted from. At the top part, where there was these empty proteins, right, there was no radiation being emitted up here because protein wasn't radiolabeled. All the radiation came from the bottom. So this kind of basically proved that DNA was the molecule that was entering a cell and transforming it. Okay? We could prove that because protein never actually entered the cell. And if you never entered the cell, there's no possible way that you can start transforming the genetics inside that cell. So we know that it is now DNA. And after this kind of experiment, this was pretty conclusive evidence. I mean, you could argue that Avery's experiment was pretty conclusive because, I mean, that's a pretty conclusive experiment as well. But the combination of both of these really solved the puzzle. And now we can stop trying to figure out whether it's protein or DNA. We know it's DNA. And now the next step is, okay, DNA is this amazing molecule. I mean, it can do everything. We got to figure out its structure. Because if we figure out its structure, we can figure out how it works, how exactly does it make protein, how exactly does it replicate, and then we can start now playing around with it. And we're going to get into that in our last chapter of, now that we understand everything about genetics, what does it do? What does it mean? What can we do with all of this information? Okay? So I hope that sorted out the Hershey and Chase experiment. Okay? Again, something that you need to really understand, as well as the Griffith and Avery experiments. Okay? So the other thing I want you to do today is to go through that video. It's a rather long video. You can maybe break it into 2 parts. You don't need to hand in that question sheet, but the question sheet is there for basically kind of making sure that you understand what happened in the video. Okay, and kind of recalling. So when you look over that question sheet, you're like, okay, I remember the video. The big point of the video is understanding the contributions of Rosalind Franklin and the fact that nothing was done by an individual scientist. Although Watson and Crick got all the credit, okay, you should note of how many other scientists contributed to it. It wasn't just Watson and Crick. It was Rosalind Franklin and Chargaff and Gosling and Pauling and all the other people that contributed to it. Okay, and that was the big point that I wanted you to get across from that video. So I hope you have a good day. I'll be doing a little bit of a recap coming up as to key things that you need to know, and then we're going to be moving on into structure, and then into replication, and then into protein synthesis. And then once we're done that, we've got all the background on information on genetics, and then we can start going into the technology and talk about, well, how do they do DNA fingerprinting? How do they do parental testing? All of that kind of stuff. Okay.